Hello everyone, and welcome to this short video from Twist Bioscience to get started with cfDNA. Circulating cell-free DNA, or cfDNA, is DNA fragments found in the bloodstream, originating from both healthy and cancerous cells. When it originates from tumors, we call it circulating tumor DNA, or ctDNA. ctDNA carries crucial information about the tumor, reflecting various genetic and epigenetic alterations such as point mutations, insertions, deletions, translocations, copy number variations, and methylation changes. These fragments are released from cancerous tissue into the bloodstream, making a blood sample a powerful, non-invasive diagnostic tool for cancer research. The ability to detect and analyze ctDNA in plasma opens up numerous clinical applications related to cancer. From early cancer detection and screening, through monitoring treatment response and minimal residual disease, to recurrence monitoring and understanding resistance mechanisms, ctDNA provides real-time insights into tumor dynamics. However, working with ctDNA presents significant challenges. Its concentration in the bloodstream is incredibly low, often less than 1% of total cfDNA. Additionally, ctDNA fragments are often highly degraded and shorter than normal cfDNA fragments, typically falling within a target range of less than 150 base pairs. The very low CT abundance makes NGS the ideal method due to its high sensitivity and ability to detect low-frequency alleles. The conversion rate, that is, the amount of unique molecules that will be converted into libraries for sequencing, is critical for accurate detection of somatic variants in DNA, especially with low-input ctDNA. Imagine trying to find a needle in a haystack. The lower the mutation frequency, the fewer copies of that variant exist. As illustrated in the table, detecting a mutation with a frequency of 0.1% from just 10 nanogram of input DNA means finding only 3 copies. Poor conversion efficiency from imperfect end repair and ligation can lead to loss of these critical rare ctDNA fragments, resulting in false negative results. This is where the Twist cfDNA library prep kit excels. We offer advanced conversion of cfDNA samples, providing up to 2 times higher conversion rates than competitive kits. This exceptional library conversion leads to lower failure rates, fewer reruns, and significantly improved accuracy. Furthermore, from even very low input, as low as 1 nanogram, our kit delivers robust rare variant detection with up to 2 times higher target coverage. This means you can confidently capture rare variants across variable and low input samples, which is crucial for demanding applications. Our library prep workflow can be completed in around 2 hours and is easy to automate on any NGS liquid handling system. Our comprehensive development process, featuring improved enzymes like our internally developed T4 DNA ligase and optimized buffer formulations, directly translates to superior performance. This results in up to 2 times higher library prep yield compared to similar on-market low-input kits, as well as more efficient conversion of starting molecules into sequence-ready libraries, leading to better data and increased confidence in your results. Our solutions at Twist offers a low error rate, especially when using UMIs. Our improved conversion rate leads to higher mean target coverage and advanced library complexity. This higher complexity, even with duplex error correction, provides superior mean coverage compared to competing kits without error correction. Ultimately, this means better sensitivity and greater confidence in your variant calling, reducing false negatives and allowing you to find those critical rare variants. The Twist cfDNA end-to-end workflow is designed for robust and reliable performance. It includes key steps like end repair, A-tailing, UMI adapter ligation, and UDI primer PCR amplification to create your libraries. Our Twist cfDNA target enrichment protocol then takes these libraries through blocking and hybridization with Twist double-stranded DNA probes, followed by streptavidin bead capture and stringent washes. This workflow delivers excellent uniformity, increased on-target rates, and is fully automatable, ensuring cost-efficient and even coverage for a broad range of panels, including our fixed panels, custom panels, and spike-ins. Let's take a closer look at the Twist cfDNA library prep workflow. It's a very streamlined and efficient process, which takes around 2.5 hours, including a combined end repair and A-tailing step, UMI adapter ligation with our re-engineered cfDNA ligase, and the PCR amplification with our UDI adapters, followed by purification and QC. The resulting libraries preserves the fragment size distribution characteristic to cfDNA, as shown in the bioanalyzer traces. The workflow can of course be automated. When the libraries have been generated, the target enrichment workflow will take them through an overnight incubation step with your probe panel and different types of blockers so that the probes will bind specifically to your region of interest. The hybridized targets are then bound to streptavidin beads and are going through a series of stringency washes, aiming to retain only the fragments of interest while getting rid of everything else. After post-capture PCR amplification, purification and quality control, the enriched libraries are ready for sequencing. This entire target enrichment process takes about 3 hours after the overnight hybridization and can be fully automated like the library prep. Twist offers a comprehensive portfolio of products to support your entire NGS workflow. Our portfolio includes essential components like Twist cfDNA pan-cancer reference standards for robust validation, Twist UMI adapters and UDI adapters for multiplexing and error correction, and the core cfDNA library prep kit itself. We also provide the entire target enrichment workflow and a wide array of panels, including our fixed panels, MRD, and custom panels, as well as you have the option to spike in any additional content with a spike-in panel. The kits are available in both 16 and 96 sample configurations to meet your throughput needs. The Twist cfDNA library prep kit is incredibly versatile and especially well suited for difficult samples with very low input DNA. It's ideal for cfDNA derived from plasma and other body fluids, as well as FFPE tissue and other highly degraded sample types. Our solutions support critical applications such as early cancer detection, MRD, PCR-free whole genome sequencing with our full-length UDI adapters, as well as non-invasive prenatal testing. Our UMI adapter system specifically enhances sensitivity for rare variant detection, enabling confident distinction of low-frequency variants in cancer research by reducing false positives. We highly encourage you to visit the Twist Bioscience website for detailed protocols and a wealth of additional resources like webinars, product sheets, troubleshooting videos, and technical notes. In summary, the Twist cfDNA library prep kit with its Twist-engineered cfDNA ligase offers advanced conversion and robust performance with low input samples as well as with highly degraded samples, empowering your research with greater sensitivity and accuracy. The improved enzymes and optimized buffer formulation enhance performance and makes the kit an excellent approach for liquid biopsies and mutation detection with low amounts of input DNA. The workflow is fast and efficient, automation-friendly, and can be combined with the Twist target enrichment workflow and any of our Twist panels. Thank you so much for your time and attention today. We hope this has been informative. If you have any further questions, please don't hesitate to reach out to us. We are here to support you in every way possible.